CRISPR, or clustered regularly interspaced short palindromic repeats, is a revolutionary gene editing tool that has the potential to transform the agriculture industry. This innovative technology allows scientists and researchers to precisely target and alter specific genes within an organism's DNA, paving the way for the development of crops that are more resistant to pests and diseases, require less water and fertilizers, and have a longer shelf life. CRISPR works by targeting specific sequences of DNA within an organism's genome and making precise changes to those sequences. The process begins with the identification of the specific gene or genes that are to be edited. Once the target gene has been identified, a small piece of RNA, known as a guide RNA, is designed to bind to the target gene. The guide RNA is then combined with an enzyme called Cas9, which functions like molecular scissors to cut the DNA at the specific location targeted by the guide RNA. Once the DNA has been cut, the cell's natural repair mechanisms are activated, and the DNA is either repaired or replaced with a new sequence of DNA. The new sequence can be one that is naturally found in the organism, or it can be a synthetic sequence that has been designed to introduce a specific change or trait into the organism. In the case of agriculture, CRISPR can be used to modify the genes of crops in order to enhance specific traits, such as pest resistance, drought tolerance, or nutritional content. For example, scientists might use CRISPR to introduce a gene from a pest-resistant plant into a crop species that is prone to pest damage. This could result in a crop that is more resistant to pests and requires fewer pesticides to protect it. Similarly, CRISPR could be used to modify the genes of a crop to make it more drought-tolerant or to enhance its nutritional content by increasing its levels of certain vitamins or minerals. CRISPR has the potential to greatly enhance the resilience of crops, making them better able to withstand extreme weather conditions, pests, and diseases. By modifying the genes of crops, scientists can create varieties that are more resistant to drought, flooding, and high temperatures, which can significantly reduce crop losses and improve food security. For example, CRISPR has been used to develop maize varieties that are more drought-tolerant. Maize is a major food crop that is grown around the world, and drought is a major factor that can reduce crop yields. By introducing genes from drought-tolerant plants into corn seeds, scientists have been able to create maize varieties that are able to survive prolonged periods of drought. This could have a significant impact on food security in areas that are prone to drought and reduce the risk of crop failure. In addition to improving drought tolerance, CRISPR can also be used to enhance the resilience of crops to other extreme weather conditions. For example, scientists have used CRISPR to develop rice varieties that are more resistant to flooding. Flooding is a major threat to rice crops, and it can cause significant losses for farmers. By creating rice varieties that are more resistant to flooding, CRISPR has the potential to improve food security and reduce the risk of crop failure in areas that are prone to flooding. Furthermore, CRISPR can also be used to boost yields. Scientists have used CRISPR technology to develop crops that are more resistant to pests and diseases, which can greatly reduce the need for pesticides and herbicides. This not only reduces the environmental impact of agriculture, but it can also reduce production costs for farmers and increase their profits. Moreover, CRISPR can be used to improve the nutritional content of crops by increasing the levels of certain vitamins or minerals. This can have a significant impact on public health, particularly in areas where malnutrition is a problem. While the benefits of CRISPR in agriculture are significant, there are also concerns about the risks and unintended consequences of gene editing. One concern is the possibility of the technology to introduce unintended changes or mutations into the genome of an organism. While the technology is highly precise, there is always the risk of off-target effects, which could result in unexpected changes to the genome. There are also ethical and social concerns to consider when it comes to CRISPR and agriculture. Many individuals have expressed their worries about the potential for gene-edited crops to be used to create genetically modified organisms, that is, GMOs, which may be perceived as unnatural or harmful. There are also concerns about the use of CRISPR to create crops that are patented or owned by a single company, which could have implications for farmers and food security. Nonetheless, the bottom line is that CRISPR is likely to revolutionize the agriculture industry and enhance food security around the world. By creating crops that are more resilient, have higher yields, and have improved nutritional content, the prospects of CRISPR in addressing some of the major challenges facing the agriculture industry today are high. It is, however, essential that the use of CRISPR in agriculture be carefully regulated and monitored to ensure that it is used in a responsible and ethical manner. It is imperative to consider the possible risks and unintended consequences of gene editing, and to ensure that any gene-edited crops that are developed are safe for consumption and do not have negative impacts on the environment. What are your thoughts about this subject? Let us know in the comments.